The kickoff of the Oceans Alive seminar series. This is a seminar series as the Woods Hole Oceanographic Institution Sea Grant program sponsors every spring. And we always kick off the series with lectures by the recent science fair winners from both Falmouth High School and Falmouth Academy. I'm Judy McDowell. I'm the director of the Woods Hole Sea Grant program, and Tracy Crago and Sherry DeRosa stopped you inside the door to hand you a schedule. And I encourage you to take the schedule home with you because for all of the Tuesdays in April, with the exception of April 16th, which is school vacation week, we have a series of panel discussions on coastal issues. On April 9th, next Tuesday, a panel of experts, including scientists, citizen volunteers, town managers, and regional planners, will discuss the topic coastal ponds, management and effects of nutrient inputs. This is a very timely topic for Falmouth as well as other coastal communities, and I think you'll find the discussion quite lively and somewhat controversial at times. But tonight we're here to congratulate 4 of the top science fair winners in our community. And we're going to alternate between the winners from Falmouth High School and the winners from Falmouth Academy. And so Luke has graciously agreed to go first. Luke Sunquest is the son of Eric and Holly Sunquest of Falmouth. He's a junior at Falmouth High School, and he won a second prize at the Falmouth High School Fair for his project entitled The Effects of Crystal Growth Rate on X-ray Diffraction Patterns. He received a third prize at the regional fair, and he'll be going on to the state fair at MIT in May. So Luke, the podium is yours. Yep. And here, I gotta put this. You can put that in your pocket. Cool. Right, just put this in. This is so you can be recorded for perpetuity.
Does it work? Is this a microphone? Okay. Hi, I'm Luke Sunquest. Project was entitled The Effects of Different Crystal Growth Rates on X-ray Diffraction Patterns, and I guess first I'll just give you a little background on my project. Almost all solids are made up from crystals on the atomic level. There are a couple exceptions, a couple metals, and glass being the major exceptions. Through the years, it's been discovered that one of the most accurate ways of determining a compound's identity is through this X-ray diffraction analysis. When a crystal acts as a diffraction grating, an X-ray diffracts off the crystal at angles which are unique for every crystalline substance. Thus, the graphs produced are a type of fingerprint for each compound. And sometimes these X-ray diffraction patterns can be the sole manner of determining the identity of a substance. So I found it necessary to learn which was the most precise way of growing these crystals for this type of analysis, since many times the crystals need to be grown to perform the procedure. I hypothesized that crystals grown more slowly would form a more detailed lattice, which is the arrangement that the atoms form together, than the crystals which were grown fast. Therefore, I assumed that the results which I got for the slow growth would be closer to the standard graphs in the textbooks, per se. The two methods that I used, the first was slow evaporation. It's pretty much your standard way of growing crystals. Supersaturated solutions were made for 3 compounds, potassium ferrocyanide, sodium nitrate, and potassium chlorate. I got these compounds just from crystal books. They said these would be some of the easiest ones to know. And I got access to these chemicals in Falmouth High School, and I mixed up a supersaturated solution at a high temperature. Once it cooled and the solubility point went down, some of the compound settled at the bottom, and I poured the liquid off the top, which was a mixture of the solute and solvent, which was water. And I consider that to be saturated, meaning it was holding as much of the solute as possible at that given temperature. After this, I put about a quarter of this solution aside, sealed the 3/4, which I had saved, and left the quarter of the solution out overnight to let some of the solvent evaporate. When the water evaporated, the solute stayed in the petri dish, which it was sitting in, and little crystals began to form. The crystals were then hung from a rod in the rest of the solution, the 3/4 of the solution, and let sit for, I'd say, about a week out in the open. As the water evaporated off the top of the solution, the solute gathered to the seed, which was hung, and formed more crystals as less solvent was available to dissolve it, so it precipitated out. And after this, I dried these crystals and saved them for analysis. And the fast growth was about the same. To start out, I made a supersaturated solution at a high temperature, but instead of letting it cool gradually, I put it in plastic flasks and sealed them and put them in an ice water bath, which drastically changed the temperature of the solution. Therefore, the solubility point went down quickly, and again, the crystals precipitated out of the solution. I then poured the liquid off the top again, stamped out the crystals, and let them dry. So now I had 3 different kinds of crystals for each method, slow and fast. This is when I got to the X-ray diffraction portion. For this method, I obtained use of a powder diffractometer at the United States Geological Survey on the Quissit campus in Woods Hole. And I had to grind up the crystals very painstakingly through a sieve and into slides. The slides were open-faced, they were metal, they were about the size of maybe $50, and when I ground up the crystals, I brushed them through a sieve or a filter, which would only let the smallest pieces go through. So in a sense, and so that's how I got the powder. I packed the powder down with a glass slide, and then removed the top glass slide, and leaving just in a flat surface of powder. This is just a little explanation of how the diffractometer works. When the crystal slide was placed into the diffracting chamber, the X-ray came in and hit the crystal. These flat layers are the different layers of the crystal lattice. It would be deflected up to a sensor, which would measure the intensity of the X-ray. Depending on whether or not the peaks, which are these lines on the X-ray, if they lined up as in this bottom graph, there would be a higher intensity, which would lead to a higher peak on the line graphs, which would come out. X-rays are used for this process because they have just about the right wavelength, so it makes observable patterns. Let me just show these graphs. The graphs that I got came out like this. This is just potassium ferrocyanide. The x-axis is degrees in terms of 2 theta, which is shown in Bragg's law equation. It's down on the bottom. It's that angle right there. It rotated the crystal from 2 to 70 degrees in terms of this angle right here, and that was considered an adequate amount of data to make the fingerprint. So that's what the x-axis is. The y-axis is peak intensity. So you can see the main peak is where the most intensity of the X-ray deflected off at that angle. Once I got these graphs, I thought the way I could determine their accuracy would be to compare both the fast and slow growth and see if there weren't some differences between the fast and slow in terms of detail. These are two segments from the graph I just showed you in a much more detailed manner. This is the fast growth. Let's see if I can get both of these on here. They're both the same compounds, potassium ferrocyanide. The top graph is the slow growth, and the bottom graph is the fast growth. As you can see, the first peak, peak number 12 right here, came out to be 2-pronged for the slow growth, but only 1-pronged for the fast growth. I could interpret this two ways. Either that there was an error leading to 2 peaks on this, or these 2 peaks were emitted right here. When I went back to my references and checked, there are supposed to be 2 prongs here. So I considered that because the crystals grown quickly, since they were grown quickly, an error occurred in the formation of the lattice, which led to the lack of detail between the 2-prong and the 1-prong. Also, over on this series of peaks, you can see for the slow there are 4 peaks right there, but for the fast there are only 3. Again, I checked this with my references, and it showed that the last peak on the right was supposed to be in the pattern, which again led me to believe that the fast growth again had an error in its formation because of the rapidness of its growth. Probably just an impurity got caught, or there was a hole filled with a foreign substance in the formation of the crystal. These are the potassium chlorate graphs, and as you can see, put slow on the top again. The top is the slow for potassium chlorate, and I determined that again it is more detailed because the peaks tend to be much more sharper and defined for the slow growth and the fast growth in this series. And pretty much this was indicative the whole way through. So my first conclusion that I drew was that my hypothesis was correct in that fast growth had less detailed patterns than the slow growth because of the quickness of its growth, leading to impurities being caught in the lattice, etc. I wanted to determine also if the relative peak heights changed between different substances. If I could universally classify, say, fast growth had higher peak heights than the slow growth in all examples. What I found really, though, was that there was no correlation for this between the substances I experimented with. So you can see by this graph, the bottom is the slow peak heights, and the y-axis is the control and the fast, which pretty much echo each other. The control was just done with straight reagent out of the can. So if the peak heights were the same, they would all line up along this line on this graph. For this particular one, which was potassium chlorate, you can see that the fast and control groups had higher peak heights. When I moved on to sodium nitrate, there really wasn't a correlation. It seemed to be spread out, but more or less around the line. And the potassium ferrocyanide also seemed to be spread out, but more or less along the line. So I determined that universally, through all crystals, you can't really classify whether or not fast growth or slow growth will determine relative peak heights in these graphs, but future experimentation, I would assume that crystals which had similar lattices, lattice shapes to the ones I experimented would have similar results in these experiments, and through further experimentation I could classify these classes into saying that chlorates have higher peaks for fast growth and slow growth, but you couldn't do that for all compounds. So that's about it. Are there any questions?
Questions for Lou?
Okay.
Excuse me, how long have you been working on this?
I started the actual research probably before Christmas. I did most of my experimentations during February vacation and pretty much worked up to the date of the Falmouth High School Science Fair, which was in early March. Yeah.
What would you do next?
As I said, I would try to work with fine compounds similar to these, instead of potassium chlorate, maybe sodium chlorate, and see, I could run my initial experiments and just to check up to make sure that the slow growth was still more detailed than the fast growth, and also try to make classifications, links between the different compounds and their relative peak heights in relation to the fast and slow growth.
Try to do different, instead of just having fast growth and slow growth, try to do something
Yeah, there are many methods for growing crystals. I was thinking you can grow them in silica gel also, which has been found to be a detailed way of growing crystals. And there are scores of ways of growing crystals, so I could also experiment with that.
Okay, our next speaker is William Gordon, who's a freshman at Falmouth Academy, and he took the top honors at Falmouth Academy's Science Fair for his project called Search for Antibiotics, Phase 2. William also placed second at the regional fair, and he'll be going on to the state fair in May. Okay, she's
Okay, my project was entitled Search for Antibiotics in Marine Organisms. And which one of these do I use? Right, left one? Okay. And I got my idea because I was just looking through a Time magazine article, and I read about how all the different bacteria that we have now are starting to become resistant to the different antibiotics that we have. And when one becomes resistant, it can lead to millions that are resistant. So I thought it might be interesting to try and see if I could find anything locally at my beach, which I have. Here, I worked at Seagull Beach in South Yarmouth, and what I did is I just went along the beach for my first phase and collected as many different types of organisms as I could find. I did mussels and all different types of seaweeds. And from here, I developed a bioassay to test out my different materials. And what it was is I took chocolate agar plates and I punched a hole out of the middle where I would then place the ground up organism. And I also had 200 microliters of different human pathogens spread onto the plates. One plate had Staphylococcus aureus, one plate had E. coli, and one plate had a yeast, Candida albicans. And from there, I went on to test it. I tried it out first on just some different known antibiotics to make sure that it worked. And here it had an effect on the E. coli and the Staph aureus, but it was not affecting the yeast. So here I've proved that my bioassay worked. And from there, I went on to test actual organisms. This is rockweed. And here you can see that the rockweed had no effect whatsoever on any of the 3 pathogens. And then I went on to test more, and this is this is of the Polysiphonia genus. It is Polysiphonia nigens, and it had an effect on the Staphylococcus aureus. To tell if something had an effect, you look for a clear ring around the material where the antibiotic has spread into the plate and stopped or killed the bacteria from growing. The results of my phase 1 test, I tested 68 different organisms, including bacteria from the sand and some small mollusks and a lot of different seaweeds, and I found only 3 to have a positive antibiotic effect. And then I continued on this year with my phase 2 by taking 2 species that I could find regularly of Polysiphonia that were both positive, Polysiphonia denudata and Polysiphonia nigens. This is a close-up picture of the denudata. I was able to have access to magnification cameras at the Cape Cod Hospital, and here you see the different plant, and the green at the bottom is codium or green fleece, and I found through identification that the denudata is parasitic and it latches on to such things. And here's a picture of Polysiphonia nigens up close, and it is usually found floating in large clumps or attached to rocks along the beach. Then I discussed this with some of my teachers, and I consulted with senior scientist at Marine Biological Laboratories, Dr. Norman Wainwright. And through him, I was able to get access to a lot of old books, and I did literature research back through the early 1900s, and I found that around the 1950s, they did some research with related species of Polysiphonia, and they found that the antibiotics within them were phenolic compounds. So from there, I was able to use phenolic, ways to take out phenolic compounds to get my antibiotic. This is just a description of my methods. First, I would disrupt the seaweed in my mom's blender, and then I would centrifuge it out to separate the seaweed, the heavier materials, from the lighter molecules that were inside it. And by using my bioassay on that, I found that the lighter materials, or the supernatant, had a much greater antibiotic effect than the heavier materials. And from there, I was able to do a solvent extraction with butanol, which I found from the literature research I did. I was able to extract phenolic compounds. And from there, it was concentrated and dried. And then from that, I used a process called thin layer chromatography, which this is a picture of the butanol sample after it's been mixed through. And here I'm doing the butanol sample. What I did for the butanol sample was you take a measure out of the supernatant or pellet, whatever you're using, and you mix in between 20 and 30 microliters of it, and then you mix it up and you let it separate. And then on the left, the green is the stuff that's been extracted by the butanol, and that is what I would test. And then here on the right, I have it dried out and concentrated. And then from there, I was able to do thin layer chromatography, which you take the dried stuff, you mix it with a little bit of chemical to get it wet again, and then you place it on a silica gel plate. And from there, you put it in a glass jar, into a solvent to extract your antibiotic, and I used one for phenolic compounds. And then this is the way it looks afterwards, and by looking at it under UV light, you can see the different separations. What this does is it separates out the different chemicals by their mobility rates and how fast they move. And you can see the different chlorophyll layers that spread out and some of the keratines. Here, the large blotch on the right is just straight phenols. Then the one in the middle is with the butanol extraction, and then the one on the far right is with an ethanol extraction. I found with the Dano data that it was about even no matter what I did, and I was able to separate out about 8 different chemicals. Sometimes there was an extra layer in between 4 and 5, but usually this was about what it was. And then for the niagens, I also found around 8 different layers, and they all traveled relatively the same amount, even if some of them were taller. Here I just show an average of the two, and even though they're far apart, they do show that there's the same amount of layers, which relates them to the same species, and also the fact that they all go up about the same, even if one of them's double. From this phase, I found that my procedure works and my bioassays are correct. I've been able to determine that both species are similar in compounds, but I need to do further testing with my butanol extractions. So far, I have not yet been able to prove which compound extracted from the butanol is giving off the antibiotic effect, and I'm also trying an ethanol extraction, which was also found in my literature research, to try and separate it out even further. That's basically it.
Questions for William?
This brings back memories to me when I was a young boy, younger than you, in Situate, and I used to go, I summer down there, sea moss up from the rocks, and I'd gather it, but I never did know what they did with it. I used to turn it over to the old fishermen, and they'd dry it out on the canvases. But I never did follow that line at all anymore, but it's just something to do when I was a youngster.
Yeah, a lot of different pharmaceutical companies within the past 30, 40 years have started to collect things from the ocean, do deep sea vessels, because the ocean is a relatively unexplored resource, and it's just amazing how many different things we'll be able to find from it.
We take it off the rocks.
Mm-hmm. Yeah, a lot of different species come into a bay and then attach to the rocks for easy access, which is helpful.
So a lot of the harvesting of Irish moss in New England was to extract carrageenan, which is in most ice creams, most paint, most cold cream, and lots of other products, and has been for quite some time.
Way back this is, though.
Yeah, but even, I mean, for at least 40, 50 years.
Yeah, seaweeds have been used for lots of stuff.
Other questions? Thank you, William. Our next speaker this evening is David McElroy, and David won the first prize at Falmouth High School Fair. He's a junior, and his project, The Effects of Copper Sulfate on the Development of Sea Urchin Embryos.
Good evening. My name is David McElroy, and tonight I'm going to tell you about the research I've been doing on the effect of copper on the fertilization of sea urchin embryos. And I've been doing this as a part of my science project with the help of some of my teachers and Dr. Robert Silber. I've been working in his lab at the MBL. In our environment, we deal with many chemicals that can be toxic, and some are beneficial and necessary while others are harmful. And some are beneficial only in small concentrations that are found naturally in our environment. One example of this is copper, and copper is necessary for many life functions. For example, the formation of collagen fibers, which are the proteins that are the basis of bone, tendons, and skin. And small amounts of copper are necessary for the proper assembly of these fibers. but I wanted to know what would occur if the levels of copper became greater than those found in the environment, and could we test for the increase of copper? And I decided to try and develop a test using sea urchin embryos as a model system. And this is like the general summary of the procedure I used. And what I'd do was I'd collect the eggs and sperm from the adult urchins, and then I'd expose the eggs in seawater to the varying concentrations of copper sulfate. And after 10 minutes of exposure, I'd fertilize the eggs and observe fertilization, and then I'd find the percent fertilized. And the first experiment I did was to find the optimal time for fertilization after spawning. And I found that there was greater percent of fertilization at 40 minutes and 120 minutes after spawning, and there was less minutes, 60 to 80 minutes after spawning. And based on this, I decided to use 40 minutes as the time to use in my experiment after spawning. And so the second experiment I did was to determine the effect of the copper. And the varying concentrations I used were 1, 3, 5, 10, and 30 micromolar copper sulfate. And I found that the 1 micromole concentration was close to or the same as the control, and that the 3 micromole just began to start affecting the fertilization. And so I concluded that that was near the threshold where the 1 micromole had no effect. And the 5 micromole was getting more than half the population, inhibiting fertilization. And by the time I reached 30 micromoles, there was complete inhibition of fertilization. And the third experiment I did, because I found a lot of irregularity in my data, and I thought this might be linked to the fact that my sample sizes weren't being controlled. And so I did a test to test the effect of varying amounts of eggs. And using concentrated eggs and then using serial dilutions, I diluted the eggs to 2x, 1x, half x, quarter x, eighth x being that much less, and then I had a control with no copper, and all the other ones were at the same concentration of copper, 5 micromoles. And I began to see a trend. You can see the control here, and then the 2x, the 1x, quarter x, and eighth x, and there's kind of a trend forming, except for the half x doesn't exactly fit the pattern. I think this might be an anomaly. And so in these experiments, I wanted to determine if copper had an effect. And this kind of summarizes my conclusions. I found the optimal time for fertilization, that copper does inhibit or eliminate fertilization, and that the copper effect is dose dependent, and that the copper effect is dependent on the number of eggs. And these results indicate that sea urchin fertilization may be a useful indicator of the presence of toxic chemicals such as copper in the marine environment. And that's about it, and I just want to thank you for your time. Are there any questions?
Mentioned earlier that they did also want a third prize at the regional fair going on in the questions for David?
Do you think the form of the copper that's toxic is the form that's added as the ionic form of copper, or do you think it's interacting with some of the organics?
Do you mean the sulfate? It could be the effect of the sulfate, but I had to find a solution of copper that would dissolve in water. So.
I was thinking more of the copper itself as an ion once it's dissolved. Sometimes it's associates with other things that are in the water.
You mean the salt? Or, I'm not sure I understand your question. I mean, okay, okay. Any other questions? Yeah? It comes from things like copper pipes, pressure-treated wood, bottom paints. Copper bottom paints are being banned because of their effects on the environment.
One of your low concentrations, the half-X dosage, where you seem to have similar results in control, that's actually been observed in the scientific literature with low levels of copper, that they actually have somewhat of a stimulatory effect. So you might look at some of those publications because that might be a real effect. It's called a process called hormesis, where the cell overactivates because of the stress.
Yeah, because I ran that experiment because I thought that, like, with the fewer cells, there was more copper per cell, and so there was a greater effect with fewer eggs.
The last speaker this evening is who also won first place at Sunquest Academy, won 70 place at the regional fair, will also be going on to the state fair. And the title of her project is The Acute Effects of Trichloroethylene on the Brine Shrimp Artemia Selena. We'll give the microphone back
how do you turn this on? The orange? Ah, okay. All right. My name is Theresa Hong. I'm a junior at Falmouth Academy. And I did my project to find the acute and chronic toxic effects of trichloroethane and trichloroethylene on Artemia selena, which are also known as brine shrimp. The acute toxic effects are the immediate effects of or the immediate toxic effects, and chronic toxic effects are the overall long-term effects. Now, the Massachusetts military reservation used to use a solvent called trichloroethylene to degrease all the metals on their airplanes. And they use this in massive amounts, and they don't use it anymore. But right now, the environment is beginning to feel some of the effects. And what this solvent does is once it hits the ground, it doesn't like to stick to the soil, so it goes right through the ground, and it's been seeping into the groundwater around here. And right now, environmentalists are trying to find out if it's been leaking into the ponds and lakes around the area. And environmentalists around Cape Cod, Buzzards Bay area, have detected plumes around the area with high levels of trichloroethylene, higher than what the government allows. Let's see. Many articles have been written on this problem because very limited data exists on the effects of trichloroethylene on Artemia. And I found this quote here in the newspaper. It says, "There are significant marine sources at risk here. This is both an environmental and economic issue. People's livelihoods are at risk, along with the environment itself, because of the apparently unchecked march of this plume to the sea." This was said by Trudy Cox, who is the Massachusetts Environmental Affairs Secretary. Before I began my experiments, as I was searching for previous literature that had been published on similar studies, I found no articles, to my knowledge, I found no articles on the effects of trichloroethylene on Artemia, and I couldn't find any articles on the chronic toxic effects, which are the overall long-term effects of trichloroethylene on any saltwater organism. So to my knowledge, and the scientists here who helped me out, this project will be providing some of the first research on this area. And let's see, I chose Artemia because they're a saltwater organism. They're about 1/3 to half an inch long, and they play a very important part of the small crustaceans group in the marine food chain. And this small crustaceans group, they play the vital role in linking this phytoplankton/algae group to these other groups here. If anything happens to this small crustaceans group, then it could disrupt the entire food chain, and it would just be really bad. And let's see. So for my project, I not only wanted to find the effects of trichloroethylene on Artemia, I wanted to compare it to another compound. Both these compounds are in the same family. They have pretty much similar properties. The major difference is that trichloroethylene is 5 times more soluble in water than trichloroethane, right here. So what I thought would happen was that I would prove that the trichloroethylene was more toxic to adult Artemia than trichloroethane because of its higher solubility rate. So I worked here at Redfield Laboratories in Michael Moore's lab, and for acute toxicity, which is the immediate effects of trichloroethylene and trichloroethane on brine shrimp, I tested 7 different concentrations of control at regular seawater: 10 parts per billion, 100 parts per billion, 1 part per million, 10 parts per million, 100 parts per million, and 1 part per thousand. For each concentration, for each compound, I had 3 test tubes. In each of those test tubes, I had 20 adult Artemia. Every 24 hours for 4 days, I counted the number of surviving Artemia. Let's see. And here's a graph of my acute toxicity data. And these lines here show the average number of Artemia surviving over this 4-day period, the average number after 24 hours, 48 hours, and so on. And as you can see here, this is for the trichloroethane, which is the less soluble compound. At the lowest concentration of 10 parts per billion, you don't see any major difference between the control. However, for the next highest concentration of 100 parts per billion, you see this gap here, which shows that it's beginning to have some effect on the mortality rate, and so on and so on. And 2 out of the 7 concentrations I tested here reached LC50s, which is when 50% or more of the population dies, which is along this line right here. 2 of them reached LC50s, while 2 of the other concentrations closely approached them. However, for the trichloroethylene, which was the more soluble compound, you see this big gap here in between the lowest concentration of 10 parts per billion in the control, and so on, which supports my hypothesis saying that the trichloroethylene was more toxic because of its higher solubility rate. And 4 out of the 7 concentrations reached LC50s, which is, again, when 50% or more of the population dies. So that supported my hypothesis. And also, I thought that both of these compounds are volatile organic compounds, which mean that they evaporate into the air really quickly within the first 24 hours. I thought that the most toxic effects would take place within this time. So here's a graph showing the average number of Artemia dead per day. This blue right here is the average number that died after 24 hours. Pink is after 48 hours, and so on. For the trichloroethane, which is, again, the less soluble compound, you see that the majority of all the mortalities took place within the first day. And same for trichloroethylene. The majority of them took place within the first day, which, again, supports my hypothesis saying that the most toxic effects take place within the first 24 hours. So next, after finding the acute toxic effects, I wanted to find the chronic toxic effects, which are the long-term overall effects of these compounds on brine shrimp. And I used the same number of concentrations that I used for acute toxicity, same number of brine shrimp. The major difference, the only difference was that instead of starting out with adult artemia, I started out with brine shrimp eggs. So I had 3 flasks, and in each flask I placed 20 brine shrimp eggs, and every 24 hours for 10 days, I counted the number of artemia eggs that were hatched or surviving, that were partially hatched and unhatched and dead. So here's a graph here. This is for the trichloroethylene. This is the average number of artemia that hatched and that are surviving over this 10-day period. For the trichloroethane, right here, which was, again, the less soluble compound, you see that they hatch, but then they die at an alarming rate here. However, for the trichloroethylene, this was really surprising. I found that they hatched, but they did not die as quickly as they did for the trichloroethane. And this really puzzled me at first because I thought that the trichloroethylene would be more toxic because it was more soluble. But I came up with a theory, and what I think happened was that since the trichloroethane was non-soluble in water, it floated on top of the water, as did the brine shrimp eggs. So for the trichloroethane, the eggs were almost completely submerged in this compound, whereas for the trichloroethylene, they weren't. So let's see. The standard deviations range from 0 to about 5 here. And let's see. So basically what I found was for acute toxicity, which is, again, the immediate effects on adult artemia, I found that trichloroethylene was more lethal to the adult artemia than the trichloroethane because of its higher solubility rate. And as my hypothesis stated, the most mortalities took place within the first 24 hours because both compounds are volatile, which means that they evaporate into the air quickly within the time. And for chronic toxicity, the trichloroethane, which is less soluble, was more toxic to the artemia eggs because it floated on top, as did the brine shrimp eggs. And the trichloroethane caused a shorter lifespan than the trichloroethylene. So that's about it.
You said that there were no data that you could find in the literature for marine organisms.
Right.
But there were data for freshwater organisms?
Yeah, there was.
How did your results compare with that? Were there any experiments like that?
There were experiments done on freshwater organisms, and there were I found one article on the acute toxic effects of trichloroethylene on saltwater organisms, and my data basically paralleled what I found. And they all died really quickly, more rapidly than they should have. And I found no data for chronic toxicity.
Well, please join me in congratulating all the science fair winners in the